Well, hi again, Mr. Bleeker here. We'll kick in on our last topic in biochemistry, which is proteins and nucleic acids. Now, if you haven't heard of nucleic acids before, think genetics, think genetic code. You've probably heard of DNA. We'll also be looking at its cousin RNA. That topic gets focused on in much more detail in the subsequent chapter. But in this chapter, we'll introduce them as molecules and discuss in general what they do. There's a lot here about protein, so we really need to get to it. Okay, head on over to eCampus and again click on Lessons Biochemistry, which will bring you to, I've already opened it here. There we go. Our specific page for this topic. We've completed these items. Now we're down to DNA and protein. We've got some good material for you. And this is an ever-growing list. YouTube video there on the structure of protein. Come on down here to amino acids, protein, and we'll look at the essential amino acids, talk about why they're essential, and so much more. So we'd better get to it. I'm going to fly in here. There we go. Awesome. So protein. Now protein comes in many forms. There's protein in animal forms, protein in plant forms, protein in fungal forms. It's an essential nutrient of life. In fact, it's a scaffolding protein. We're made up of protein, and that's what our genetic code encodes for. So let's look a little bit closer. What is it? Well, we've been studying carbohydrates and lipids and things like that. So not surprised to see carbon, hydrogen, and oxygen. But now what we'll kick in a little bit more here is nitrogen and sulfur. So in a typical dry weight of an organism, you can think of a mummified organism if you will, it's half of our body weight. In fact, protein is all that our DNA encodes for. So if you look at protein, you can look at muscle fiber, structure of the eye. Your hair is entirely composed of protein. Your fingernails, keratin protein. Hemoglobin, you look at the structure in your red blood cells that carries oxygen, that's a protein. So it's just so many things that it actually does. And of course, very important component of our cell walls, regulating things that come and go, and all of our enzymes are proteins, running our metabolism. So something we generally cannot do without. Now, throughout this unit, we've been talking about Lego pieces. I always love to refer to monomers as pieces of Lego that you build up. It's a good analogy. Well, the polymer is the buildup, and the monomer is the individual piece. So when you look at a protein, look at a piece of hair, for example, it's made up of many little subunits that we call amino acids. There we go. And they're the monomers. And as we bring them together, you're going to see that trick that you've seen with synthesis in other biological molecules, specifically the removal of water to build up long amino acid chains. Now, when amino acids come together, what we refer to that as a peptide bond. It would be easier, wouldn't it, if they would call it an amino acid bond, but they don't. They're called peptides. So long strains of amino acids are called polypeptides. These bonds are between nonmetals. We're looking at covalent bonds. They're very strong, and they give three-dimensional structure to our molecules. So does a certain degree of hydrogen bonding, which we'll talk about a little bit later. Now, as far as the different kinds of amino acids, there are really 20 different amino acids in total, but there's 9 that our body can't make. And I always find that interesting. It's not surprising we don't have all the genetic coding to synthesize what we need. And if you look at our pets, for example, they have different coding. For example, cats and dogs can make vitamin C. Now, that's not an amino acid, but they have the genetic capability to put together that molecule. We just don't have the ability to put together everything. So we can formulate many amino acids, but we have to get 9, and sort of debatable. Sometimes they talk about 9, 10 essential amino acids that have to come from our diet. Now, these names are kind of interesting. When you look at them in tables, they usually only use the first portion. And I'll just do a little highlighting here. Histidine, for example, you'll just see represented with the first three letters. Isoleucine, leucine, lysine, methionine, phenylalanine, and the list goes on. Tryptophan, there's a nice little one that you might have heard of. Tryptophan, warm up a little bit of milk before you go to bed, eat some turkey around Thanksgiving, and that's the amino acid that can help us feel very sleepy lying on the floor after we've had way too much to eat. Doesn't sound familiar at all, does it? When you remember the main elements in amino acids, just remember this. Chons. Worked for me, will work for you. Because that comes up all the time. What are the main elements that formulate amino acids? Carbon, hydrogen, oxygen, nitrogen, and sulfur. I'm just going to pop out of AirPlay for just 1 second. There we go. I always find it easier to leave an app with AirPlay. It's hard to pull that tray up sometimes. Okay. I just have to remember. Here's a nice little table. So obtained from nutrition, right? We can see it's highlighted in red here. The ones that are essential, the ones that we have to get from basically from our diet. And then there's the non-essential amino acids that can be synthesized by the body. Think about this. When you consume protein and it's reassembled to make your hair or your nails, or you're lifting weights at the gym and your muscle mass is increasing, that protein that you consume, your digestive system, it's almost like a large sort of Lego castle-like structure. And when it's broken down, it's broken into all the little Lego bits. Well, those Lego bits are amino acids. And based on your genetic code, based on your DNA, those little pieces of Lego, those amino acids, are put into new orders, new sequences, if you will. And depending on the sequence, it can make many different things in your body, from heart cells to liver tissue to hair coming out of the top of your head. So it's important to get a well-balanced diet and to make sure you have the amino acids fully in supply. You want to have good proteins, healthy existence. Okay. Just going to pop out, go back to our mirroring session. There we go. Now, as far as the amino acid structure, if we look at one piece of Lego, there's a few things that you'll see in amino acids. Now, you can see this written one of two ways. I want to clarify this a little bit. Actually, let's just pull this down just a bit like that. So you'll have this primary carbon atom. And remember that carbon shares with, covalently with, it needs 4 electrons to get to the end of the periodic table to be electrically stable. Its outer shell would be very happy. Now, generally that primary carbon, let's see here, we'll just draw a partnership with hydrogen, kind of par for the course. On the end, you can also see the carboxyl group drawn that way. One thing to be aware of is that that hydrogen right there is sometimes a little bit fragile, a little bit loose. So you might see that it flies off into solution. So when you look at the carboxyl group, focus on the carbon double bonded to the oxygen and single bonded to another oxygen. That hydrogen's a little bit optional there. It sort of depends on the pH of the solution that the amino acid finds itself in. If you've studied chemistry at all, it's sort of related to the topic of buffers, but we'll get to that later. Now, here's where the amino acid goes to its other primary group. It's being a pain. I'll give you a nitrogen. There we go. And this is called an amino group. Now, there's room for 2 nitrogens, but every now and then it'll take on a third. And you see the end is a little bit more positively charged. Right? If you take on hydrogen is essentially a proton. So if under different sort of pH levels it takes on an additional hydrogen, that end would be sort of positively charged, And this end, if it lost a proton, would be a little bit deficient, right? It would be a little bit negatively charged. Here's what's really important. I'm going to switch. You need to know the functional ends. So you've got an amino group on that end. You've got a carboxyl group on this end. You have your central carbon. What gives the identity to each amino acid isn't just those things. Those things make it an amino acid. But this R group, and I always think of a good way to remember it is the remainder. This guy down here can vary quite wildly. And there's a whole side chain of atoms that will build off that side, and there's many different amino acids to look like. Or, sorry, to look at. They don't look alike, is what I was trying to point out. Let's pop out of this for just a second. Let's pop out. There we go. And look at what some of the amino acids, what their appearance is. Let's see here. And I thought I had that tab up. That's okay. Now, it's fairly quick. If you Google amino acids, I'd meant to do this earlier, but it won't take me very long. The different amino acids, you can call up a chart, and lots of different ones. So let's choose a nice color one here. There we go. Now, when you look at the R group that can go off of different amino acids, there's many different structures. You can see some are quite short, some are quite long. This is arginine, histidine, lysine, aspartic acid, glutamic acid. So take a look at a few of those. Oops. I'd already gone there. Let's go back. This. There we go. That's a nice sort of overview here. And I'll just zoom in on that. There we go. Now, if we look at the amino acids, ones that are polar, non-polar, for example, some are electrically neutral, some are charged, have different polarity on the ends. But really what I want to point out is just look at what's going off of these different three-dimensional structures here. Look at tyrosine, for example, asparginine. And these give different chemical characteristics or different amino acid identities, is I guess I'd be more correct in saying, to our different aminos. So there you go. It's quite an assortment. Just pop back here. There we go. Now what becomes quite interesting is how do we take these individual amino acids and follow the genetic code, right, of our DNA and put together hair fiber, for example? Or how do we put together Rod's cones at the back of your eye? Well, according to the DNA sequence, the little Lego pieces go in different orders. I mean, that's an oversimplification, but that's literally what's happening. Now, it comes down to what we've already seen before, which is condensation synthesis, the removal of water to bring these molecules together, or the introduction of water to break things apart. In this case, you've got your carboxyl group, often called your acidic group, and you've got your amino group here, sort of oriented head to tail if you would think about it that way. And again, we have that reactive hydroxyl group and one of the hydrogens going off, the amino group. Now, the removal of those for condensation synthesis produces water, one molecule of water. And what we get when we bring these together is we get a direct link between the carboxyl's carbon and the nitrogen of the amino group. And that gets a special name. It's called a peptide bond. And as we bond many peptides together, what we get is a polypeptide. So introducing water to bring the amino acids, or removal of water to bring the amino acids together by removing it, we call that condensation or dehydration synthesis. Alternatively, introducing water at this point, right here, if we add a water molecule, what we can do is cleave that peptide bond and go back in the other direction. So it's fully reversible. Neat little animation for you to take a look at. It's straight off the course site. Let's see if I can. There we go. That was actually efficient this time. And here you go. Here's a peptide bond. Carboxyl groups, amino groups, and the introduction of the two is generally facilitated by an enzyme, and there's your peptide bond, denoted in red. It's good to watch it a few times, you get the idea, but you've seen this from our other examples. Now we have enzymes that do this, biological catalysts that orient these molecules towards each other, in basically improving the odds of the chemical reaction occurring. In this case, dehydration, aka condensation synthesis. Pretty neat little example there. And of course, that's on the course site. You can look at that till your eyes bleed. That's it there, but sorry, no animation here, so I thought I'd pop out. There you go. So your amino acid structure is just a little bit more on that. For example, there's alanine. And if you study, when you begin to study a little bit more biochemistry here, that can be written a couple of ways. A CH3 group, sometimes called a methyl group, right? That's more of its accurate name. Now you're seeing it drawn two ways. The way on this side here, that's sort of what we call empirical. It doesn't give as much information about the three-dimensional structure, showing how the bonds, the angularity is really formed. This is a little bit more accurate on the left. Takes up more space, so it's empirical versus biochemical. Anyways, it's alanine. Okay, here's valine, and you can look at it. Sometimes a little bit easier to look at the empirical structure. It's just a little bit more condensed. Thank you for that. I have a hair appointment. And you can see this ring here of carbon atoms, a couple of methyl groups there. And in each case, if you look, notice that carbon always is always sharing in a way where it gets 4 electrons. Case in point, 1, 2, 3, 4. It should take you back to your Lewis dot diagrams, and that information is very important. You always want to retain that. Cross-curricular. Pop through, here's phenylalanine, right? So they're saying that the R group can have a ring structure? Absolutely. And these can become quite long side chains. But the important point is that you always observe, there you go, amino group, carboxyl group, your primary carbon there, and it's that off which your R group is founded. Now, this one is extremely important to know. And year after year, when we go over this topic, we talk about amino acids, and each of these little balls here is an amino acid. So for the purposes of illustration, we start to just draw them as little structures. Now, a primary amino acid, the first thing that you would get if you line up amino acids head to tail, head to tail, head to tail, is you would get a long string of them. This looks like a bead necklace. That's the first thing that will form, but it's not the last, because you have to consider there aren't just covalent bonds involved in amino acid structures. When you look at oxygen, it's a very electronegative atom, and it likes to it's a little bit of an electron hoarder. And if oxygen, for example, sees hydrogen just a little distance away, it will pull on it, and you get a that little pull is a hydrogen bond. Now, kind of zoom in on this just a little bit. There we go. I just have to. If you notice this nitrogen right here, it's also like oxygen. It's very electronegative. It's hoarding electrons. So hydrogen itself will be a little bit positive. When you see hydrogen bonding or hydrogen forming covalent bonds with other things, sharesies, you always have to remember that hydrogen is not very electronegative. It really doesn't get its proportion of electrons. It doesn't get to hang on to them very often. And as a result, hydrogen will be a little bit positive. So hydrogen being a little bit positive is going to be attractive to adjacent or nearby electronegative atoms. And right up here, what do we see? We see oxygen. So oxygen itself is a little bit electronegative. So you can think of it as just being a little bit negative like that. And when you see hydrogen nearby, it's a little bit positive. And that's the whole reason that you'll get these hydrogen bonds, drawn as dots in this case, between nearby hydrogens and oxygens. And what this begins to do is it's sort of a sticky bond, and it will cause the amino acids, adjacent chains of amino acids, to come a little bit closer together. And it starts to account for some of the different shapes that we begin to see. They'll become sticky. Now, they can become sticky in sheets. And a beta-pleated sheet, in this case, is like amino acids amino acid chains that are starting to link up side by side. It's not fully accurate to draw it this way, but sort of get the idea. They're sort of hooking up to each other. And when you look at something like a beta-pleated sheet, what you're looking at, a really great example of that, is silk. And silk is incredibly strong. Why? Well, it's covalently bonded. That's one thing. It also has a lot of these little hydrogen bonds that help to hold it together. Now, when you look at hair structure, it's also, if you take pull one hair out of your head, it won't be too bad. Just go for one and grab it from end to end and pull. And you notice it's extremely elastic. It stretches. Now you wonder, well, what accounts for the elasticity? Well, let's zoom in on this guy for a little bit. Unlike silk, if we look at a hair fiber, it forms a twist. Now that twist gets a name. It's called a helix. And it's springy. If you look there, you'll see that the chains or the peptides will look down at adjacent peptides and they'll hydrogen bond. So instead of it being sort of a long sheet, what it's doing is it's curling up. And what's doing the curling, if you look at this amino acid here, for example, it's being attracted to an adjacent amino acid. So when you look at the carboxyl group of one amino acid, it may be interested in the amino group much further down. Well, it's all about electronegativity. The oxygen is very interested in an adjacent hydrogen. And what it's doing is that's basically like a little sort of static bond, and it pulls up the molecule so that it starts to form this sort of helix. That's a very interesting little structure. So you don't stay with your primary structure. Primary would be like the first thing that would be formed. Now, by primary structure, we'd say primary or first degree. Now, what would happen due to the hydrogen bonding nature of amino acids is they'll start to form they can form these secondary structures, these beta-pleated sheets, right, like silk, or they can form alpha helixes, like we see in our hair. That would be the second thing, or the secondary structure. Now, this doesn't exist in isolation. When we look at the expression of our DNA, we don't just get these things. In fact, it can go further. And I'll always like this animation here. If we get lots of alpha helixes, these alpha helices can start to form in sort of ribbons. And what you get, if you get enough of these ribbons together and there's adjacent hydrogen bonding and the ribbons are forming beside each other, what you'll see is you'll get something called a tertiary structure. It's like a ribbon glob. At this point, we would just call it a glob. It's the third thing that can happen. So tertiary, or third degree structure. Now, if we have globs, that's not to say that we can't keep going with globs and compound globs onto globs onto globs, etc. I like to call them super globs. An example of this would be the production of insulin. Insulin itself begins as a first degree structure, and then we get the formation of helices, and then we get lots of helices forming globs, and we get globs upon globs upon globs, and that takes us to the fourth degree structure, which is our quaternary structure. So insulin is like this. So is hemoglobin, a very large proteinaceous molecule. Now that's an interesting point I raised. At what point are we talking about a protein? Well, when we start to get a polypeptide chain, that's by definition a protein. So an individual amino acid, you could look at it and say, hey, is that a protein? I'd say, well, no, not really. We need to get a couple of them together. And there you go. So here you go. Just the information portion. Really, when we're looking at the peptide bonds, remember, we're talking about covalent bonds. Really doesn't assume anything about hydrogen bonding, which is what brings these additional structures together. There's a little. There we go. I'll just give you a minute, scribble a few things down here. I think I've referred to this quite a bit. One thing we didn't talk about, I didn't focus on a lot, is there's also a lot of sulfur in the structure of several of these amino acids, and you can also get interactions between the sulfur molecules, and they call it disulfide bridges. I didn't focus on it too heavily, but it's just another thing that can cause your structures to curl up. Just one more attraction. And when you look at the structure of insulin, you start to get this superglob, and you can see the ribbons and all the different structures. It's quite complex. It takes a lot of amino acids, if you think about it, one amino acid at a time, absolutely. And we put this large molecule together. Now, if you don't remember what insulin does, it is the only hormone in your body that can take sugar out of your blood and store it as starch. Now, that's animal starch, so we're talking about glycogen. So it's the only hormone that can take sugar out of the blood and put it into your muscles and into your liver, which is pretty important, right? You want to save your sugar and metabolically distribute it to the body as you need to. Interesting thing about insulin, if you've ever felt that sinking feeling after lunch, right? Where you are so tired that you can't listen to anyone teach or talk, for that matter. This molecule can take the sugar out of the blood very effectively. In fact, it can do it too effectively, and you're left with too little sugar in your blood, and your brain begins to run on less, and it shuts down a little bit, and you feel tired, and you want to have a nap. True story. Neat thing about proteins that you can do with them, they will denature. I've got an animation for you to help explain this. When you cook a protein, the structure is a little bit scrambled, a little bit rearranged. And I want to show that to you now. It will appear, oops, it will appear sort of like, for example, when you cook eggs, it'll get a white appearance where previously the egg white, that protein, was clear. And it's worthwhile taking a look at the explanation of that. I think, there we go. Frying an egg. So we'll step through this just sort of a piece at a time. So you can denature a protein with chemicals, right? Strong acid or a strong base. One of the most common ways to denature a protein is to fry it. Radiation will also do a number on it. So there you go. Egg white, 85% water, a little bit of protein in there. The egg yolk itself, of course, much higher in lipid content. I just prefer to scramble my eggs. That way my kids don't fight over what portion of the egg they don't want to eat. Now, when you think of the amino acid side chains, as soon as you begin to cook it, the bonds are going to get a little bit scrambled, a little bit sort of rearranged, and there's a little unraveling that goes on. And what you start to get is you still have these, think of them as stringy sections, right, of interconnected proteins, but what they're doing is they're rearranging and they're making these little sort of thick pockets. And as the egg hardens, light really can't get through that. So you've scrambled the arrangement of the chains and you've cooked the egg. So the proteins unfold a little bit, come into new sort of interconnected arrangements, and you've got a white fried egg. Pretty cool. Now you know what was happening when you were denaturing your egg for breakfast this morning. Now, denaturing your proteins can be quite dangerous. Think, for example, humans, when we get a fever, we don't really want our temperature, where it starts to get lethal, we're 97 degrees naturally, where it starts to get lethal is around 104 and 105 degrees Fahrenheit. That's when seizures can occur. Our bodies drive up our temperature to make it possible for our immune cells to get into regions of our bodies where they need to fight the good fight. The problem is that temperature can go too high. My children, for example, will reliably barf on command at about 101 degrees. That temperature commands it. A low-grade fever is just natural. The body's fighting off infection. But a compounding high-grade fever, that's when you might remember your parents giving you acetaminophen, also known as Tylenol, because it has an ability to bring the fever down. Right? But fever is not the enemy, but it can denature proteins. Not good. Now, you've probably seen this before, and you'll say, hey, look, there's DNA. A double helix. I mean, you saw a single helix before when we were looking at alpha helices, right? Looking at hair, and that was the, let's think about it for a second. Wait, secondary structure of DNA. Now, nucleic acids, like DNA, are sources of genetic information. What's interesting is that one of those nucleic acids, and there are 4 nucleic acids, adenine, guanine, cytosine, run out of space, and thymine. Let's finish that off. There we go. Oh, that is a terrible E, but okay. I'll fix the C as penance. There we go. Those are the 4 primary DNA. Well, let's actually back up. We'll call those the 4 primary nucleic acids, okay, that are associated with DNA. Now, it turns out one of them is also sort of a dual agent. It also, in the body, becomes a really great energy storage molecule. And we'll cover that in a second. See if you know which one. Now, you might have covered this in grade 11. We used to cover this back in grade 10. Sort of depends on your experience. These bases are, if you think about DNA, we always talk about what is the moniker, what is the moniker, what is the monomer of DNA. This molecule up here, right, is a polymer. There's lots and lots and lots of them there. So yeah, you're looking at chains and chains and chains and additions there. Now in this case, you're looking at one of the monomers. So the monomers are nucleic acids. And that monomer has 3 parts. 2 parts are structural. They just give it integrity, strength. In DNA, they make up the edges of the ladder. So we've got the phosphate group and a 5-carbon sugar. Now that's like the edge, but that's not the genetic information. Instead, you've got these you see these nitrogen rings up here. So we say, of course, it's nitrogenous, and that becomes the base of the genetic code. That's the base of the information. Now, I've had students, admittedly, in the past say, why do you call it a nucleic acid and then call that a nitrogenous base? What they mean by nitrogenous base is it's the base of the genetic code. We're not talking acids and bases there, so don't confuse those two ideas. This is what is read as your genetic code. The other 2 parts are just structural. Okay, let's expand on this a little bit. There's 2 types of nucleic acids, and it's not just DNA. You might have heard of RNA. In Biology 11, we talk about that as being the genetic code of many viruses, including HIV. Other famous RNA viruses would be the cold viruses, many of the flu viruses. There are viruses, many viruses out there that also have DNA as their genetic code, but the big difference, I'll just draw it like this, is that if these are the little genetic bases, the adenines and guanines and such, RNA is older. It's a little bit more primitive, and it's a single-stranded molecule. Whereas DNA, now I'll draw it unzipped, not as a twisty ladder. DNA will put its nitrogenous bases in towards the middle. This is a single-stranded molecule, whereas this is a double-stranded molecule. Now, admittedly, RNA itself is more mutable. A lot of viruses keep ahead of our immune systems because that molecule can mutate quite a bit. Whereas DNA, organisms that use it, it's quite conservative. It doesn't mutate at such a high rate as you see RNA will. Now, they're both genetic codes, admittedly. However, we need RNA for more things than just being a sort of a genetic code. In fact, your ribosomes are made up of that. So let's say exactly what RNA is. So you've got ribosomal RNA. That's why we say RNA. So we'll say ribose for short. Now we also have 2 other forms of DNA, RNA. We have transfer RNA and messenger RNA, which we'll look at in this course. And those are very important as helpers to DNA. In fact, you need all 3 of the players that I just showed there to make proteins in your body at all. So RNA is not just a genetic code for some of the more primitive organisms, it's absolutely essential to make proteins in our body. DNA is definitely a master blueprint. But without those forms of RNA over there, we can't express DNA. A couple little things about DNA. They refer to it as deoxyribonucleic acid. And it's deoxy. Well, okay, deoxyribonucleic. See why we use three letters? Especially on an iPad writing with the stylus. There we go. And I'll just pause there and I'll say, wait a minute. Deoxyribo? That tells me pretty well all I need to know. If you look at the five-sided sugar that forms, of course, part of the structure of the edges of the ladder, and you look here at the second carbon, now remember, this is the first carbon here, second, third, fourth, and there's your fifth carbon up here, which actually leads over to your phosphate group. Often the way we draw that is just a quick little drawing here. Go like this, and we'll draw the phosphate kind of coming up from the side here like that, and then we draw the nitrogenous base like this. You'll often see this drawn. And this is your sugar here. It's DNA is different than RNA. DNA has taken an oxygen away from the second carbon in the ribose sugar. So I'll just undo that. So that's the difference right there. DNA has deoxygenated the second carbon, whereas ribose is fully oxygenated. And you can see that right down here. There you go. So, deoxy versus oxy. Now you know. There are chemical families when you study genetic information, you study DNA, and you'll often, whenever I introduce DNA, I always refer to it in this order. You say A, G, C, T. And what you've got are the purines and the pyrimidines. Now, starting out with the letter A, that's sort of the first thing in the alphabet, shortest thing, right? The shorter word is purines. That's how I've always remembered it. And if you can remember that the short word goes with the shortest letter in the alphabet, the first one, you'll remember it. A, G, C, T. Now, these are double ringed. So that's an important point. And the pyrimidines, well, they have a single ring structure. Now, there is a sneaky one being mentioned down here. Right here. Uracil. And you're wondering, hey, why haven't you written in U? Well, I tend to focus on studying DNA first. What you need to know about in RNA does not use thymine. Ever. No. RNA's gone with an older version. Think of it that way. It's gone with uracil. So whenever you look at RNA, you can think about it this way. I'm going to go like this. If this is DNA's series of bases, then RNA, sure, it has adenine, and it has guanine. It's all good. It has cytosine, but then, whoop, there you go. It's got uracil. And that's interesting. That comes up in our study of DNA, and we use a purple color to represent it just so that nobody forgets it. Not to worry. We'll cover it in detail. Here are the nitrogenous bases. When you look at thymine and cytosine, you can see the carbon rings and the nitrogenous structures in there. That's why we call them nitrogenous. So there's their structures. So the pyrimidines are single ringed, and they fit, sort of lock in step, key-like fit, I guess, with their corresponding pyrimidines. Now, you might notice how they're lined up. Here's a little rule. Adenine itself will always bond with with thymine, and it actually makes a double bond. So we'll go like this. There you go. We'll look at it in the reverse. There you go. So A bonds with T. Now, if we look at cytosine, they form triple bonds, as it turns out. There you go. Oh, I almost had it. There we go. So adenine bonds with thymine, cytosine bonds with guanine. Now, in the case of RNA, just put a little. Oops, I don't need the tray, I just need a line. Now, this is our DNA circumstance. All you have to remember, the difference, draw a little lightning line here. The only difference in RNA is that adenine bonds with uracil. Uracil bonds with adenine. And that there's no difference over here. RNA follows these rules. Here, cytosine, guanine, they bond as per normal. Just that one distinction to remember. Here you go. The bonds, look at them, are hydrogen bonds. Now, you may have thought they were covalent bonds, but I thought I'd wait till we got here to show you this. Again, electronegativity, right? When you look at nitrogen, it's fairly electronegative. It's going to be liking an adjacent hydrogen over there, and that oxygen is very much attracted to the hydrogen you can see on the left in the first example. And in this case, sort of look at this row here, guanine, and you'll see that we have an opportunity for a triplet hydrogen bond here. And this hydrogen bonding is very important, and I'll give you a reason for that. Your DNA itself is looks like a twisted ladder. Well, we'll study in the next chapter, it unwinds and becomes sort of this really long ladder. We open up sections of it, and it is absolutely vital that we be able to disrupt these bonds and reform them. Let's grab a nice green color. We need to be able to get in here and pull this apart and read one side or the other. So we will make messages from our DNA, looking at one side or the other, and then when that's all done, we need to let the molecule heal or seal back over. And it's a fabulous mechanism, the way that our genetic code is expressed. And it comes right down to such simplicity of hydrogen bonding. We can get in there with enzymes, unwind DNA, make a copy of its message, and then wind it back up and get out of Dodge. And there you go. So you can see how it's a twisted ladder, right? We're looking at DNA in this case. It's twisted in these directions, and that twisting saves an incredible amount of space. It's the ultimate in file compression. However, when we go to read a section of it, we have to untwist it. And now we can see the edges here. There's the repetition, right, of our phosphate, or the phosphate group with a sugar that's adjacent, right, a ribose sugar. And you can see what it does. This sort of regular bonding. I tend to just draw it like this, just a little bit easier, right? You get the idea. And that covalent bonding is quite strong and gives the edges a lot of integrity. But we can't have strong bonds between our nucleic acids in the middle. If our nucleic acid had bonds like that, we would never be able to read the genetic sequence of this molecule, and it would just be self-defeating. We wouldn't have evolved, we wouldn't have survived. The DNA molecule will look like this. There's your twisted ladder. The edges right here, what they're trying to convey to you, is that you've got your phosphate group and your deoxyribose sugar there, right? That's the edge. The genetic information, right? Your nucleic acids are in the middle. Okay, so just a brief mention here about RNA. First thing I notice, there's uracil, right? There's no thymine. No T. So, there you go. Oh, by rungs of the ladder, that's come up a couple of times. By rungs of the ladder, what they mean are these, right? As if, and I'll just write right over top of it, as if these would be the steps that you would stand on going up the ladder. That's what they mean by rungs. Now you know. Okay. I mentioned that one of those molecules, okay, adenine, was a double agent. It did a couple of things in the body. Turns out to be a really interesting molecule. Now, if you look at battery technology, phosphorus itself, let's stop and think about this. Phosphorus makes some pretty interesting compounds. Now that's the phosphate ion, and it's capable of exchanging. It's got a valence of 3 minus. You think about it, it's got electrons to share. It's fairly interesting. 3 electrons. So it's quite an energetic molecule. What would happen if we took the basic nucleic acid, adenine, and we tacked on not just one phosphate group? Like when you look at the structure of DNA, it has one, but it's using it structurally. What would happen though? Let's just undo that. If we made 3 of them. And we connected a triplet. What do you have? You have an extremely high energy covalent bond. And this, because there are 3 phosphates, you have your foundational molecule, right? Now, in this case, we refer to it as adenosine, right? Adenine, you get the idea. Go like this. Like, oh, wow, that's adenosine triphosphate. So it's got 3 high energy bonds between its phosphates that can be broken. And in fact, this is what is made by your mitochondria. Mitochondria, through a process, now if you go further in biology, you'll study something called the Krebs cycle, and there's more to it than that, but through a series of enzymatic reactions on the membranes of the mitochondria, remember we always draw it like this, right? Through a series of reactions in the mitochondrial membrane, or on it, you produce very high energy molecules known as ATP. And this is the true energy. This is the electricity that runs the place, if you want to think of it that way. And as it's broken down, it's sort of like a chemical battery. Let's draw it like this. This would be good. Yeah, we'll go with that. ATP, if you want to think of it, is like a full battery. And as each phosphate is cleaved off for the purpose of metabolism, right, the energy state is lowered a little bit. That would be like ADP. Now there's just 2 remaining. And adenosine monophosphate, like this, is when we just have like almost like a singular nucleic acid, as if, and I'll just, adenosine monophosphate would just be that, just the singular phosphate in that case. Lost what I wrote. There we go. So, as it turns out, one nucleic acid makes a very good energy storage molecule. And that process going on in the mitochondrion, known as cellular respiration, well, what happens? Really, we feed, it's a little bit of a simplification, but once we feed glucose, broken down, mind you, because it's too big to go in as a 6-carbon, it gets broken down in little 3-carbon chunks, and once it's fed into our mitochondrion, well, out pops this molecule right here, ATP. And it's amazing what can be done with this. Runs our entire system. We couldn't live without it. Not very efficiently, anyways. Now, you have to say, well, what's the source? Really, what was the source of the glucose? Where'd that come from? Because here it is. Where did we get the glucose to run that? Well, if we think about it in the purest sense, it's produced by photosynthesis. So our friend, the sun, provided all the energy that plants would ever need, providing it's sunny out, to fuel photosynthesis. And remember from previous lecture, move to a black color here. There we go. Photosynthesis goes on. Those little green pancakes, right? Those are called, that's a granum. Plural is grana. And it's in the edge of the pancake, right there, right on the membrane, which is where chlorophyll persists, and we generate glucose. Glucose is fed in little 3-carbon chunks once we break it down. In here, show you a little bit more of the story. And the process of cellular respiration produces ATP over here. And there's always a little bit of water. If you think about it, when you break down a carbohydrate, there's your source of water. So you're going to produce water. It's a little bit of a wasteful process. It's not 100% efficient, so you get a little bit of heat. So when you breathe out in the morning and it's cool and you can see those water droplets in the air, well, you can thank your mitochondria for producing those. And there we go. They've drawn their water over here, obviously, because what they're pointing out is when anything burns, you get a bit of heat, you get a bit of water, and you get carbon waste, which is perfect because this guy, our friend the chloroplast, needs CO2 to get the job done. So if we travel in space, you can see why plants make a perfect partner for us, because we're breathing out the waste product that they need, and they're emitting oxygen, which we need. We could never pack enough stored oxygen to make it to Mars, for example. We would need plants to help give us a breathable atmosphere in our vessel. We just wouldn't be able to make it otherwise. Unless they found a way of storing oxygen, which is much more efficient than anything I've ever heard. Okay, that's it. Wow, that takes us to the end. Let's see here. Yeah, definitely takes us to the end of our lesson. So we've covered a lot of ground. We've covered proteins, covered amino acids. We look at their structure. Heck, how the amino acids form peptide bonds, right? Through condensation synthesis. How we can break up long chains of peptides through hydrolysis. It's totally reversible. And we looked at the nucleic acids. Wow, we covered a lot of ground. Ladies and gentlemen, thanks for listening. And that's another one for the books. Ciao.